part of this webinar series, are you guys also doing some evaluation of the students?
Sir, I'm sorry, please, sir.
As part of this webinar series, are you guys also evaluating student participants, something like that, and based on, or it is just an educative program?
No, sir. Frankly to say, this is just an educative program. In fact, we are sending the feedback links also, sir, at the end of the program.
Okay. Because I know, like, couple of other colleges, I did a similar session. They even had, after the program and all, they had a question answer session to evaluate students, that kind of things for us.
No, sir.
Colleges were trying.
Not exactly, sir. We're not evaluating anything like that. Just we are sending some feedback regarding the session, how it has been carried out, and what are all the technical difficulties they have faced. Nothing more than that, sir. In fact, the session will be open at the end, so that any speaker have any sort of doubts, they can directly communicate with you.
Yeah, sure. That is always welcome. In fact, it's good to always have an interactive session rather than, it really doesn't make any presenter happy if it is a unimolar or unidirectional discourse.
Definitely, sir. Yes, sir.
Be an interactive, then I think everybody should take maximum, make use of such discussions.
Sure, sir. Sure, sir. Definitely, we'll see, sir. Principal sir, shall we start the program?
Yeah, Dr. Ishak, please proceed.
Yes, thank you. Good afternoon, one and all. Welcome back to second session of this international webinar on pharmacological screening methods. Today we have Dr. Srinivas sir, who is connecting all the way from Mumbai, live with you. So without any formal things, let us start the program by the introduction of Dr. Srinivas sir by Mr. Ramana Baru. Ramana Baru, over to you.
Good afternoon, everybody.
Ramana, sir.
Sir, good afternoon to all. So welcome the international webinar on pharmacological screening afternoon session. So I am going to introduce a brief profile regarding Srinivas sir. So Dr. Srinivas Gulapalli had his Bachelor of Pharmacy and a Master in Pharmacology from Andhra University, and he did his PhD in Pharmacology from Naipur, Mohali. And after that, sir, he worked as a postdoctoral researcher at Mount Sinai Medical School, and later on he has worked in new drug discovery based research for about 15 years at various positions in India in reputed pharma companies like Dr. Reddy's Research Foundations and Glenmark Research Center Mumbai and MCure Pharmaceuticals Pune. And currently he is working as a freelance expert drug discovery and pharmacology consultant for pharma industry, right? And coming to the point of patents and publications view, in patents and publications are, he has 8 product patents and 6 methods of use patients and 18 international publications and 1 national publication and several presentations at international and national forums. And sir, he worked as a general manager for new drug discovery research in MCure Pharmaceuticals Limited. So, and he also worked as a deputy general manager, that is senior principal scientist in Glenmark Pharmaceuticals Mumbai. And sir also he worked as a postdoctoral scientist at Mount Saint Medical Center, New York, USA. So this is the brief profile and introduction about Dr. Srinivas sir. So thank you, sir. Thank you.
Thank you. Thank you, Mr. Ramana. Thank you so much for introducing the today's speaker. So without any delay, let's have a lecture of Dr. Srinivas Kumar, sir. Sir, please, sir.
Yeah. description about myself, and really thank Mr. Ramana and admin and Dr. Madhusudan Shetty, and also to the management of Shanti Ramudu College of Pharmacy for giving me this opportunity to interact with you all guys on this web seminar series. Coming to okay, so today I'm going to talk on drug discovery strategies typically followed in a drug discovery industry. It can be either a small time biotech industry or a big time pharmaceutical company. So how things happen and all those things in a preclinical discovery research perspective, I would like to give, and what is the importance of drug screening and what kind of strategies one has to the industry normally adapts and implements in this process.
Sir, excuse me, sir.
Okay. Uh,
Sir, your voice is a bit less, sir. We are unable to hear you.
okay. Am I audible now?
Yeah, it is better now, sir, than previous.
Okay. Yeah. So coming to what is drug screening? Drug screening or pharmacological assays or bioassays or biological screening. Several people call in several different terminology, but all those things, if we see, drug screening typically is a process by which drugs are normally identified and optimized by by passing through several chemical compounds, or it can be a biological compound. Uh, you test them in various biological assays, and then the idea is to select a drug candidate so that the drug candidate is finally being progressed into a clinical trial. Normally this is in an industrial setup. This is a typical strategy where it depends on different industries. Small time biotech companies, they may synthesize and screen and test some thousand compounds or combine a couple of thousand compounds, whereas some big pharmaceutical companies, especially to find out the first initial drug hit, what we call it is called as hit. Any hit compound, I'd like to give some, throughout the seminar, I will be using this terminology for the benefit of all the participants. So normally a hit molecule can be anything. It can be a chemical moiety or a biological moiety. Normally it will have something like a micromolar range activity. So that means first time you are trying to find out a particular chemical scaffold, whether it is having the desired potency on a particular target system or something. So this is that. Whereas normally what happens is once having found a hit molecule, people try to optimize it and come up with more potent molecules. Most of the time, potent molecules will be they will be in a nanomolar, like 100 nanomolar, anything like 3-digit nanomolar, 2-digit nanomolar, to single-digit nanomolar potency, they will be there. So, but coming to drug discovery process itself, if we see, it's a confluence of various disciplines, such as pharmacology, biology, biochemistry, molecular biology, cellular biology, and it's a highly complicated process, and which requires various kinds of expertise and also experts in the field. And most of the times, people who are having a broader interfacial understanding are very much required in this drug discovery process. Here, I would like to highlight one particular thing for the benefit of all pharmacy graduates. There is a particular, we are all pharmacy graduates, and there is a particular terminology people use with respect to pharma students, that pharma students are like a jack of all trades, you must have heard all these things people saying, jack of all trades and master of none. Okay, at a bachelor's level, this is applicable, but as a farmer graduate, as a farmer student and graduate, as he goes higher in his education, once in a master's, where you will do a specialization, and then some people definitely go for higher education, PhD and all. So this the idea is, once you have mastered particular specializations and all, based on the specialization, everybody will have utility. Coming to drug discovery, I'll say a pharmacologist, by a pharmacologist directly will have a role, a big role to play in drug discovery. And so is the medicinal chemistry, pharmaceutical chemistry people with medicinal background, medicinal chemistry background. These two people, but apart from these two professionals, you will have utility of various other specializations. Let us say pharmaceutics people. There will be a lot of utility of pharmaceutical people in pre-formulation at a particular stage of drug discovery process, where you having found a drug candidate, a lot of pre-formulations need to be done for, to decide whether we are administering the drug in the form of a tablet, capsule, and all. So there will be utility. And coming to analytical chemistry and analytical bioanalytical people, throughout the drug discovery process, there is a heavy utility of people from pharmaceutical analysis. There will be heavy utility. So it's very important. Why I am stressing this particular aspect is, if you see my last line, broader interfacial understanding. Remember, for the entire drug discovery process, a pharma graduate is the best suited person for having that what is required as a broader interfacial understanding. So this is very important from the perspective of drug discovery. I just wanted to highlight, since I am addressing pharmacy students from your college. Hello. Yeah. So coming to the pharmacological drug screening, as I mentioned, the pharmacological drug screening normally it runs through the entire thread of this thing, and throughout the breadth of preclinical discovery and early discovery process. If we carefully look at, I will highlight, there are two different kinds of discovery processes, which are typically called, I will explain in detail in the later slides, which are typically called, what is called as, forward pharmacology or classical pharmacology, and our classical pharmacological discovery, pharmacology discovery, and another is target-based discovery. So morning we have heard Dr. Kavimani also highlighting, biological extracts, herbal extracts, and some other these things. So in olden days, most of the times, this a classical discovery research used to people used to do, where they follow, they typically take up a particular herbal extract and try to test, and where it is functional, in which particular disease area it is functional and all. But that approach is no longer is existing anywhere. It is there for particular purposes whenever people have a definite understanding or a definite utility of it, traditionally or classically used some medicinal agent, where that knowledge is there, in order to quickly test, that kind of methodology is being tested out. But otherwise, if we see, it is most of the time in the current last 2 decades or so, more than 2 decades, it is wherever it is drug discovery that is happening in the industry, most of the times it is called as the target-based discovery, where with the advancement of molecular biology, cell biology, and gene cloning techniques and all. So people first establish the role of a particular molecular target with a particular disease process, and then they will validate the target, drug discovery target, and then process goes on. So most of the times, what I will be discussing about drug discovery process, this target-based drug discovery process. So here, at even at the time of target validation, there will be a lot of testing, biological assay testings need to be done. Either it can be genetic or it can be cellular based or proteomic based studies need to be done. And so at the time of target validation and the target identification and validation also, there is a heavy role of drug screening or biological screening, bioassay screening that is there. Utility of that is very much there. So then having particular validated target, the real process of drug discovery starts from here, where companies will have compound libraries and they'll be testing several thousands of compounds in a primary screen using advanced technologies like high throughput screening and combinatorial chemistry. All these kind of things people in the industry they adapt to find out what is the best first initial hit molecule. Most of the times, people work with a different particular chemical. If it is most of the times in the chemistry, especially, I would like to mention, people work with different chemistry scaffolds because you never know which particular drug moiety or which particular chemical moiety actually works. So the people work with at least two to three different kind of chemical scaffolds people work with, because nobody knows which particular chemical scaffold will give you a lead molecule which can be further developed into a clinical program. So there you will have a lot of primary and secondary assays. So in order to develop the drug candidate, identify the drug candidate lead, hit molecule initially, and then go on to optimize the hit into a lead molecule, and then you have there are several other things that have to be done to optimize the lead molecule that you have found to finally arrive at the drug candidate. So here I would like to highlight one particular thing. A lot of people think that okay, drug screening means it happens only at the initial level, at the time of finding out a hit molecule or optimizing a lead molecule. But that is not that is not a very correct understanding, because drug screening or biological assay screening runs throughout the drug discovery process. That we will, what it is, we will come to see in later slides. So this slide actually highlights what I am trying to introduce you. The two different approaches of discovery. One is the traditional process, which is called phenotype drug discovery, or conventional pharmacology, and sometimes people also call it as a forward pharmacology. And the other one is target-based drug design. I'll come to this slide again. So this particular slide is a comparative comparison between classical approach and reverse pharmacology or target-based approach. In the classical approach or phenotype discovery, normally what happens is we'll start with some herbal extract, which in olden days it used to happen, and then try to identify the active ingredient out of that herbal extract, which is found to be active in some particular have some biological role in working in some particular disease or something like a disease, pharmacological disease model in vivo pharmacology, almond models or something, and then try to find out what is the ligand, and then having tested the extract, there can be several active components. Out of the active components, identify which particular active component is actually responsible for your biological or pharmacological action, and then try to understand that particular scale of that particular compound, and then do regular screening in various pharmacological functions, and uh, then there will be even in this particular aspect also, after once a particular active drug moiety has been found, that drug itself may not be an ideal one to develop. So then again, a role of medicinal chemistry comes, and people will try to synthesize based on that particular natural product moiety. People try to synthesize more molecules or congeners. People normally call them as congeners. So all those things again will happen, and ultimately, if people are satisfied with particular, okay, this particular modification of the original molecule is giving us excellent pharmacological actions, not only just in vitro testing, in vivo, and also from a pharmacokinetic and all. So then there in the as an end effect, the molecule actual molecular target which is responsible, that basic research happens in the end in the classical approach, whereas in the target based approach, especially the various advancements in the molecular genetics, cellular and molecular biology techniques. People, what in the last 3 decades, especially industry all over the world, what they do, they first establish a particular molecular target which has a definitive role with the disease process, which has got a significant role, and then they will try to do a lot of basic, all this happens as part of basic research, where they establish that any type of modulation of this particular target, whether it is really giving us the beneficial therapeutic efficacy or not, that they will establish in the very first place. So that is normally called as the process of target identification, drug target identification, and then drug target validation. These two things. So then you will have a molecular target. Here it can be a receptor, it can be an enzyme, it can be a kinase product, it can be an ion channel, it can be a ligand-based ion channel, or it can be a nuclear receptor, or it can be some other intracellular target which can be targeted. So having found that molecular target, then people will have a lot of compound screening. Here, it can, in the case of chemistry-based approach, it is going to be library screening. People normally call. What happens is in industry, big time industry, since they are doing this as a continuous process, they must have synthesized. They may be having thousands and thousands of compounds in their compound library as part of their continuous program. So which particular chemical moiety is the idea will be to find out that which molecule in the first place, which particular chemical moiety is the best to interact with the target of our interest. If the target, like normally when you have a target the advancement of drug target crystal structures and all these things, and molecular biology and all, people will also have, they must have already got a complete decipherment of the structure of the molecular target protein, and they may be having a crystal structure, and there will be a docking, and there's a lot of bioinformatics has brought a lot of understanding into docking, and then trying to see that utilization of virtual screens, especially where particular different classes of chemistry, classes of molecules are docked onto the crystal structure of the protein, and try to see that which has a best chance to interact with the protein. In fact, the recent COVID, you people must have seen in literature and news items, people adapting such kind of advanced techniques to understand and so this is like as a COVID crisis, as Dr. Kavimani in the morning is highlighting what is called as drug repurposing. Immediately, already established, already available drugs, whether any of the available drugs are better suited to interact with or which particular based on their mechanism of action and their location, whether they are able to interact with the SARS-CoV viral protein or not. So all that kind of things people have done that. So the idea is you have to find out a hit molecule, and then once you have a hit molecule, you optimize it into a lead molecule, and then based on that lead molecule, I will explain in later slides. Just having found a lead molecule doesn't mean anything in drug discovery. Whether that lead molecule or a lead chemical structure or a lead chemical substance, whether that is really druggable or not, whether it has druggable properties or not for further development, all these things are very, very crucial and very important. So all these things need to be kept in mind very much. So let us see in this pictorial diagram also in a this is like normally at a drug target from it in a target discovery program and a phenotypic discovery program. What happens is in the phenotypic approach, there is no knowledge of what target the particular extract or a particular, it can be a chemical substance or it can be a some active ingredient from a particular extract or herbal extract or something. Like when let us say when you have paclitaxel, a cancer molecule, or you have some other kinds of drugs, plant-based drugs, natural products. When people know that some traditional knowledge from a traditional source, a traditional Ayurvedic or some Chinese medicine source, there are already existing things that some people are using some plant extracts for a particular disease purpose. It can be Vinca rosea, Vinca alkaloids like Vinca rosea, Vincristine, Vinblastin. We all know those things are working in particular thing, but at the point of time in the initial when it is just what we know is only just this particular Ayurvedic product or some herbal extract or some it is being used in some part of a Chinese traditional medicine or Indian traditional medicine, but we don't know anything about that. So in that case, what happens is in the traditional phenotypic approach, as I have shown, all the kinds of tests are done, and ultimately we will arrive at the target, which is the molecular target for that particular extract or ingredient. Whereas in the target discovery program, from the day one we know what is the molecular target, and we are in the process of finding out which suitable chemical substances are better interact with the target protein and in a potent manner, and then try to develop those molecules. So coming to here, if we see some of the comparisons I have made in the form of a tabular, in the form of a table, where you have in the case of molecular target, yeah. Here in target-based approach, we know what is the molecular target to work with. But in phenotypic, this is something need to be identified, and most of the times it will be identified at the end of the cycle. And coming to cost, let us see the comparison. A lot of people think that phenotypic approach is simple, there is nothing, but there exists actually more cost. Target-based approach is much lower from a cost perspective, if we see. And the speed of discovery process, it's always rapid in the case of target-based program rather than phenotypic approach. And ease of SAR. I think a lot of students and the faculty, all of you will immediately correlate with what is known as a structure-activity relationship, which is very important. In chemistry, pharmaceutical chemistry, people must have, studied about that. Structure-activity relationship, just minute changes in a particular chemical substance structure can change the activity of the compound in a larger way. So doing structure activity relationship also is much easier in a target based approach rather than phenotypic approach. Because what happens is in the phenotypic approach, you are working with a particular herbal extract or something. If that ingredient, and you painstaking, you have tested it. You tested it, some extract, it is showing some pharmacological activity. And then you try to identify what is actually real active ingredient. And then if it is working, then that needs to be further. It may not be ideal candidate there, and you may have to, one may have to modify that in a further chemical modification may need to be done through to that moiety, active moiety, and then in that process you actually may lose. So any modification, whatever the active ingredient which we found out to be active, okay, but even a slight, and that is not an ideal candidate. And any slight modification of that, if it loses activity, then the whole purpose will not get served. So doing even structure activity relationship in a, kind of a research and chemistry research, doing that also is a tad bit difficult in a phenotypic approach. And so most of the time here, screening methods are most of the time target-based approach, and here it is this thing. One outcome is drug outcome. Like most of the times, when people found out paclitaxel, it is a first-in-class molecule. So you will have a chance to identify a first-in-class drug out of a phenotypic model. Whereas in a target-based approach, you can better modify it, and then you can have actually a best-in-class drug rather than just first-in-class drug. So both, at times, both may be acceptable. Even today, even though the target-based drug discovery process is very popular, but still sometimes even pharma industry and other academic institutions and universities, sometimes they even adapt, even till today, people adapt to phenotypic drug discovery based approach also to find out the first in class, a really unknown thing, that kind of thing. So the idea, both approaches may be good or bad, but the thing is, one has to decide which particular pathway is best suited, either it is an industry or some academic institutions, whoever conducting such research. It is up to them to decide which need to be taken. But one thing I want to highlight, the last point here, irrespective of which process is being done, most of the time, some kind of a drug screening in the various biological assays are very much. It's compulsory to screen molecules in various biological screening assays, and and most of the times it is this entire process is a cyclical process. People think that okay, you tested few molecules and you got a drug. That is definitely not the case. In a particular drug discovery program, typically multiple thousands of molecules need to be synthesized for optimization of its potency. And then once potency is being optimized, where you found out two to three different drug classes have been identified, and then later identify which particular drug chemical scaffold is better suited for further development. That is totally a different thing. So it is very important to have the kind of, so that is the reason why the entire drug discovery process is a cyclic, it is called a cyclical iterative process, because a medicinal chemist will synthesize, and pharmacologists and biologists they test it, and then how the structure activity is evolving, and to find out particular chemical moieties or compounds which are having a good nanomolar potency, and then again modify, and then go to the next stage to find out whether it is having solubility, whether it is having permeability or not. All these things are very important. So here, this is a typical chart which shows and highlights the importance of biological assay screening, which we are calling drug screening assays. Importance of drug screening assays in the entire thread of discovery process, in the target identification level, target validation level, and then hit discovery optimization here, where based on in the initial primary, having found out a particular target, molecular target, people screen large library of compounds. It can be thousands and sometimes lakhs of compounds people screen. That is normally big pharma do, whereas small biotech pharma and small time chemistry, small time biotech companies, pharma and biotech companies, they normally adapt to something called as pharmacophore strategy. They will somewhere in some basic research, out of some basic research or something, people will have, okay, this particular pharmacophore or a drug pharmacophore or a particular class of chemistry structure, substance based compounds are known as having known as hit molecules interacting with this particular target. And then what medicinal chemists, they do, they will do a pharmacophore based on the understanding of medicinal chemistry and this thing. They will quickly do, they will work around few types of chemistry classes, and they'll keep on modifying the structure to find out best structure activity relationship where we can find out nanomolar potency compounds. Then there comes a stage of lead identification, and then optimization. It is very important. Having found out one chemical structure having good potency doesn't mean anything. It need to have it need to have a solubility. It need to have permeability. It need to have bioavailability. All those things are normally comes in the process of optimizing the lead into a discovery model as a lead candidate for further clinical trial. Yeah. So this slide just I have this particular slide highlights various kinds of strategies people in the industry people adapt to find out that initial hit compound or hit and range, to find out the molecule which is in a nanomolar. Oh, sorry. Hello, am I audible, right?
Yes, sir, you are audible, sir.
Yeah, yeah. Okay. There is some disturbance from my side. I just wanted to cross it.
No, sir, it's audible. It's clear, sir.
Yeah. So this is this slide. You know, having we are discussing about biological assays and drug screening assays. It is very important to what kind of desired characteristics are required for this kind of drug screening assays. So the pharmacologic, the particular discovery or drug discovery, drug screening assay, whatever we are selecting, it should be pharmacologically relevant. The assay system should be pharmacologically relevant in the sense it should there should be a form the assay we are doing should be a perfect representation of the disease biology and the molecular target we are dealing with. This is very important for predictability of hit to lead molecules and further translatability of that compounds that are being found from that particular assay system. And then the next point is robustness or reproducibility of the assay. Until unless the assay is robust and reproducible, then it won't be of any use. So in any process. So for that particular purpose, the assay systems which we are working with, they should be rugged, and you should have a quality of the assay quality need to be defined with you can have a agonist based and antagonist or inhibitor based validation whether the particular assay is working, because you will have set of standard compounds which will work in that particular assay, whether they are giving predefined data in the predefined way or not. So that can be tested. The other thing is whether the assay has a compliance with the regularly used reagents. This is very important. So most of the times what happens is since people will be dealing with chemical structures, the particular chemical substance need to be solubilized. So for that one may have to use various solvents to solubilize, and then the solvent that is being used to solubilize the compound should not interfere with the assay reagents. This is very important. Most of the times, if the solvent itself is interacting with the assay reagents in a regular biochemical assay or even cell biology, cell-based assay, then it will interfere with the outcome of the assay. So this is very important. Then another important thing is assay throughput. Whether in olden days we used to do some assays, bioassays in a test tube based. So that there at a time you will be doing only a few compounds or something. Now technology has changed a lot, and now people are using 24 well plate or 96 well plate and miniaturization. In traditional times, people used to do a particular bioassay in a total volume of 10 ml, 5 ml or something. Nowadays in the industry, with the utilization of modern technology and all, people are using 96 well plate or 384 well plate, where the total assay volume is just hardly few microliters. Like it can be 500, total assay volume is 500 microliters, or even 100, 200 microliters, like that. And it is also important whether a particular assay system is adaptable to automotive. Big pharma nowadays, they do a lot of robotic automation where the entire testing defined protocols, and you have a blood analyzer, and even in India for various purposes people use automated blood analyzers, and biochemical diagnostic firms are using several automated robotic and computer assisted testing in standard formats, and where the instrumentation and all the reagents were being supplied, and because of the automation and all, they take exactly how much particular reagent need to be taken, and it will be mixed in the particular 96 well plate or 24 well plate or 384 well plate, and then ultimately the plate will be moved taken into the system instrument, and a particular a fluorescence based or UV visible absorbance or fluorescence or bioluminescence, this kind of end parameter will be this thing measured normally. So here there will be a lot of what we call as primary assays and secondary assays, and most of the times These primary assays and secondary assays normally used for conforming pharmacological assays are outcomes which we have found out in the primary assays. And some other times secondary assays, some types of secondary assays are being done to further confirm the particular mechanism of action. Some assays will be like that, and some in some of the assays will be mostly biomarker. A particular molecular target which is related to a particular disease can give rise to a biomarker. Biomarker can be anything, like HbA1c is a biomarker, or blood glucose is a biomarker. For any diabetic, in a diabetic drug program, you will have a pre-validated biomarkers, and are otherwise in a total new discovery process also. There will be some biomarker identification will be done in the first place, where the particular disease process is linked disease-based biomarkers, and whether those biomarkers are adaptable with the particular target what we are dealing with. So these things will be predetermined, and the as part. Sorry, there is some phone call. Sorry for the disturbance. So these things will be done in a predetermined fashion. So let us now look at some of the, out of the drugs that we are using, these drugs come from different particular different target classes they come from. It can be a G protein coupled receptor, it can be a nuclear hormone receptor or an ion channel and all. Based on this, I don't want to go through each and every aspect, but normally what I would like to highlight is there will be a set of either biochemical primary assays and another set of cell-based assays. Cell-based assays are secondary assays where functional. In case of radioligand, whether a particular drug is interacting with the particular G protein coupled receptor target or not. For that, you just may have to do a binding assay, and also you can do some functional assay. And then how that there can be several mechanisms that are normally associated with a particular G protein coupled receptor target. One particular second messenger system can be a cyclic AMP or a calcium evaluation or a potassium or inositol triphosphate parameter as a second messenger. Those things can be tested. So those kinds of different kind of secondary cell-based assays can be adapted. I'm just highlighting this for that purpose. And similarly, different technologies. It can be a UV visual based absorbance. It can be a luminescence, or it can be fluorescence based technology can be used for various different targets. And especially when if it is a kinase target, and nowadays a lot of kinase based drugs are there. Various technologies like FRET, which is called normally a fluorescence based resonance energy transfer technology is used, and alpha screen technology. All these kinds of different technologies are being adapted, and these are some other set of primary and secondary assays that are normally used for histone modifying drug modifying enzymes. Like we have there are so many histone based which at the level of DNA they will work, and then a transcriptional level, a particular at the level of transcription, like that means DNA and RNA at the level of transcription of the RNA into a particular target protein. At that level, if some drug target is there, so that kind of targets, and are nucleic at the level of nucleic acids. So some drugs they work at the level of DNA binding and nucleic acid level. All those things, different kind of these are just tabulated information which one can easily go through. What kind of assays, primary and secondary assays, are normally being adapted. So here, I'd like to give one specific example. It is very important to give some examples. This is a particular discovery research program which we were doing a couple of years back at MCure Pune, where we were doing identification of indolamine oxygenase enzymes and IDO and IDO-TIDO. It is called indolamine oxygenase or particular inhibitors which are being you have utility in an immuno-oncology project. So where here these are a set of we found out a particular generic structure of the generic chemical structure where R1, R2, R3, all these things are different substitutions, chemical substitutions that can be done in a typical structure activity relationship. So there to find out various properties. These are the particular C log P and log P. Log P values are normally they are very important, especially from a permeability perspective. So these were some of the things. So here, ipacadostat is actually a lead molecule which is already in a clinical development from the company called Insight. So there we are having a similar kind of a program at that point of time in discovery research, and we have our own lead-like molecule which we wanted to compare with the ipacadostat. So there for that. Believe me, for arriving at this particular lead-like molecule, we already out of 3 different chemistry scaffolds, we around synthesized around 1200 more different chemistry compounds to arrive at this particular compound, which has got somewhat similar potency in various primary assays. This is a biochemical assay where our molecule of interest is having around 320 nanomolar, and the competitor is having a 72 nanomolar, and then cell-based assays. So being an enzyme, this is an intracellular enzyme. So it is very important whether you have a right kind of parameters to do cell penetration or not. The molecules which don't have any minimum permeability, they don't cross the cell barrier and start acting the enzyme or target which is intracellular. So you need to test in a cell-based assay, and it is a recombinant target assay in a cells. And then there are physiological cancer cells, HeLa cell line. In that cell line also we have a comparative efficacy, comparative potency with that kind of this thing. So this is a different kind of an example where I have talked about G protein coupled receptors, where a lot of radioligand binding studies were done to screen compounds with either with agonist, you can have an agonist compound or an antagonist compound. So to just do this is a typical G protein coupled cannabinoid receptor program. It was this program was happening at one point of time in Glenmark Research Center, where we have a program to find out the CB1 antagonist for potential anti-obesity drugs. So there we have tested various standard compounds and also our internal compound in these binding assays to find out whether they are potent compounds against the particular binding assay which we were doing. And then these are set of antagonist compounds with our molecule also. One of the GRC molecule is a molecule where we have similar kind of potency as compared to standard compounds. So this particular work has been published in a neuropharmacology journal. So coming to, I have been highlighting the importance of optimization, what is called as a lead optimization. So you have a potent molecule that doesn't mean anything, as I said earlier. So the molecule need to have solubility. It should have a right kind of permeability. So several kinds of biological assay screens need to be designed to test these molecules. Some of them are like people will do log P or log D in a physicochemical experiment. People will find out what is the log P value and log D value, and some of the time, some nowadays various chemistry softwares are there where based on set of chemistry structure you are having, there is some prediction softwares are there which will give prediction value for log P. And most of the times, this prediction value, if for a particular compound, if you really have a very good potent molecule, and if you the same predicted value can also be tested in a lab to really see whether the log P or log D is really relevant, is comparable to the predicted value or not. And then permeability assays like CACO2 cell assay and MDR1 MDCK permeability assays. It's very important for the chemistry compounds, whatever we are dealing with, whether they are stable or not, metabolically stable or not. And all pharmacy graduates, they will appreciate the fact that first pass effect. That means if the compound is immediately getting metabolized, then it is of no utility because the drug will not be in the system for longer time, and most of the times the drug moiety may not be bioavailable also. So it is important to have some assay systems to test whether the particular chemical based substances we are dealing with in this story, whether they are metabolically stable or not. For that, you can quickly do a lot of in vitro biological assays like liver microsomes we can use, or at an advanced stage, we can even directly use hepatocytes, liver cells, and in a cultured hepatocytes, people can test whether it is really stable or not, from metabolically stable or not, by incubating the drug substance with the particular enzymes in a particular assay protocol, and to see how much of compound is remaining without getting unmetabolized after a particular 10 minutes, or different strategies people normally adapt. And then another thing is, most of the times we need to have a clean compound. In drug discovery, it is very important to have the compound should be specific and selective and specific for the therapeutic purpose which we are doing, and it should not have any add other cross target effects, and also it should not give rise to some kind of drug interaction with any other drug, because patients will be taking a lot of other drugs. So all these things need to be tested very earlier in the program, in the drug discovery program. Further, you need to do a lot of CYP450 enzymes. You will have microsomal liver microsomes in a particular 96 typical 96 well plate. You can incubate in a particular assays. You can incubate the drug with various CYP enzymes, various different kinds of CYP enzymes, and see that whether any of the known CYP enzymes are getting inhibited or getting inducted. You have most of the known CYP3A4 is a particular enzyme. Almost 50% of the drugs are getting we know that they get metabolized by CYP3A4 enzyme. And then similarly CYP2D6, 2B6, another 2C9, 2C19, various different types of CYPs are there. So it is a regular practice in the drug discovery approach to test our molecule whether it is inhibiting, because if it inhibits or it induces these enzymes, then you can potentially have a drug-drug interaction at a later stage. And the people don't want to such kind of risky things, because drug discovery process is a time consuming and it's a very cost intensive. So for these two purposes, so availability of such assays has largely simplified the drug discovery process. So this is important. And at the level of lead optimization, level, various other kind of biological assays like safety tox assays. It can be, you don't want a compound to be having cardiac toxicity. For that, you can test in cardiac myocyte cell culture, cell-based cells you can test. Or it should not have liver toxicity, or compound should not be cytotoxic or mutagenic. So various kinds of assays are there. These are the set of assays which are mostly will be utilized in the process of optimizing the lead optimization into a drug candidate, actual drug candidate, which will be taken into further development at a in a clinical trial. So the druggable lead candidate, it is important. Any drug, there is a particular thing that is normally being said. Any drug need to be, first of all, should have certain abilities. Those abilities are solubility, permeability, and a metabolic stability. And then such molecule is bound to have good bioavailability as well. Having good bioavailability is very important in development of, will greatly help developing that molecule into advanced stages. So most of the things what I have mentioned in the previous slide, it is there in the form of a table. One can quickly go through solubility assays, permeability assays, and log P, log D evaluations, microsomal stability. This is for metabolic stability, and then CYP450, CYP enzyme inhibition assays, and then hepatotoxicity and cytotoxicity kind of assays. So this is this is a typical drug program where which is being published in Cancer Research journal also. So where we have a set of parameters which we compared with our lead molecule, with our lead-like molecule, with standard comparator from a which is already in drug development, which is already in clinical development. So if we see, this is human liver microsomes. So metabolic stability, whether our molecule is stable or not. We have most of the parameters what we tested. We have comparative parameters. This is plasma protein binding. So it is very important, plasma protein binding. How much, like most of the times, what we call as a free drug and a bound drug. So any drug substance, once it goes into the blood component, if it gets bound to plasma proteins, it is of no use, because it will not participate in interaction with the molecular target, and it will not show any pharmacological efficacy. So it is important to have how much percentage free drug we are having is very important. So in order to find out a simple pharmacological bioassay can be done, which is called as the human plasma protein binding assay. In humans also we will do, and sometimes we will do based on which is our preclinical animal species which we are testing, either in mouse or rat. You will normally people tend to do rat, mouse, and also human plasma protein binding. Simple assay can be done, and most of the times the data will be of great help in further later. So finally, summary, till now what we have seen, the importance of various drug screening assays, biological assays or bioassays, whatever you call it, and the importance of drug screening in the entire thread of discovery research, right from target identification to target validation, and then hit identification, optimization of hit molecules into lead molecules, and the lead molecule and actual selection of drug candidate, which we further push that into clinical trials. So if I have to summarize all the things what I have presented till now, so the advent of various advanced technologies like cellular molecular biology, cell isolation, cell line generations, gene cloning, gene editing, genomic, proteomic technologies, in fact bioinformatics, in silico strategies where we can screen compounds in a computer assisted docking system to identify whether a particular chemical moiety is interacting with the protein crystal structure or not. All these advanced methodologies and technologies have greatly simplified an otherwise incredibly complex drug screening and drug discovery research process. And some of these recent advances in technologies have really made it possible to integrate otherwise complex and time-consuming biological assays and physiological assays into simplified formats, which can be done in a simplified manner in a 96 well plate with a very good throughput and for successful implementation of identification of drug candidates in drug discovery program. Okay, here some of the things I would like to just highlight. This is a particular this is a particular strategy framework strategy being adapted at AstraZeneca, which they published a couple of years back in Nature Reviews Drug Discovery. So for them, as per their strategy, they use one kind of a thing called as a 5-right approach. It is called 5R or 5-right framework approach. So since drug discovery is time consuming and and very cost intensive, it is very important at every level of research process. You always need to be on the right path. So based on their vast experience, they come up with this particular kind of approach or a framework strategy where you need to work with the right kind of target. Right? What is right kind of target? You can find out any molecular target, but if the molecular target doesn't have very good relevance with a particular pathological disease, then it is of no use. And then sometimes even if you are having good link of molecular target with the disease process, whether you need to see whether it is a right kind of approach, whether you have a predictive biomarkers available to better predict all these things, and then what kind of right tissues or what kind of right indication. A particular drug may have most of the times pharmacological effects in different parts of the body it may show, but which particular pharmacological activity related to which particular organ system is the best thing to find, that need to be find out for commercial utilization, commercially, in order to tap the commercial utility of that particular thing. And then right kind of safety and like efficacy is one aspect. Safety is more important. First of all, the intended drug should be safe, it should be highly safe. And then right kind of patient population. This is very important. Especially this is something happens once the drug discovery program goes into a clinical trial level. Choosing the right kind of patient is also important. And for a particular target or a particular drug, there can be several indications, and out of the 2-3 indications, which one is more right indication, especially from a commercial perspective, because industry obviously they are not doing charity. They will try to, having invested up to in a particular discovery program, they must have invested $1 billion or something. Obviously it should give some kind of returns. So right kind of commercial potential is also important. Especially, let us give one simple example. For an autoimmune drug discovery program, an autoimmune drug, it can be useful in rheumatoid arthritis. It can be useful in psoriasis and psoriatic arthritis, or any other autoimmune condition. But out of those 2-3 different pathological states, which is the best possible indication where we will have sure shot success? that kind of understanding is very important. So that is the reason why right target, right tissue, right safety, right patient, and right commercial indication. All these things are very important. So this is just I wanted to share, about a big pharma, how they think to just make everybody aware how normally pharma industry and especially big pharma they approach in dealing with a particular project in drug discovery. And this is just one slide where certain things are very important in drug discovery, which I would like to call five P's and one important thing in drug discovery. A researcher need to have passion, perseverance, perfection, poise, and practicality. And at the same time, the drug discovery team, need to be passionate. You should not get connected with emotionally and try to push any kind of a drug molecule which may not be best suited. So the some of the decision making need to be done in an impassionate manner. Obviously impassionate means I am passionate. And then for any success anywhere, irrespective whether it is a discovery research or any kind of research or even in academics, in educational this thing, focus and commitment are very important. So I thank everyone for your attention, for patiently hearing through the session is open for questions. Sorry, I think in between, I think video has gone. Yeah, session is open for interaction, question answer interaction. Please.
Sir, good afternoon, sir.
Yeah.
Sir, I am Shivashankar Reddy from Shantaram College of Pharmacy.
Okay.
Sir, I have a small doubt.
Yeah.
So in preliminary stage, if a synthetic compound has got some antibiotic activity.
Yes. Okay.
And it is having MIC value is very low, and it has got a good antibacterial activity to a particular organism.
Okay.
And in the next step or in the process of drug discovery,
yeah,
should we go to the pharmacokinetic and pharmacokinetic studies, or should we still establish the model of this antibiotic activity?
See, it can be anything. Like you may be dealing with a particular. It can be a herbal extract, or it can be a synthetic chemistry compound, right?
Yes.
You will do this, you will be doing some microbiological assays you have tested. And if the molecule is good potent, but that is what I try to explain. Like you can, normally people what they will try to do is, in different assays, like these antimicrobiological different species, whether they'll be testing different strains of bacteria and all those things. All those people will normally try to do water. See, normally what happens is, even in an antimicrobial thing, having efficacy like one particular strain of bacteria may not be sufficient, because there are already so many antibiotics, and you normally people look out for broad spectrum antimicrobials rather than having efficacy in a particular strain of bacteria, a particular bacterial species. People will try to look out for broad spectrum, whether it is having efficacy in both gram positive and gram negative strains or not. So that kind of initial screening is very essential. And if you have good potency, let us say some molecule is having a good nanomolar potency in a suitable way, which is suitable for a broad spectrum this thing, then you need to understand whether that particular drug, that particular chemical substance which you have tested, that may not be the ideal candidate to develop into a drug. Because once testing in a petri dish and petri plate, it is a simple procedure. But once you administer that in the form of a drug, it has to go into the system. It should have bioavailability, and all those aspects need to be looked at, right? So until unless the drug doesn't have bioavailability, then it won't be of any use. Either you give it through a pill or through an injection. Once it goes into the biological system, if it is getting metabolized, but that particular, in a petri dish, it may be working effectively. But if we administer ultimately to a human, it should be stable. All the optimization, like pharmacokinetic, what we normally call ADME means absorption, distribution, metabolism, and elimination. So this is called as something like in a DMPK, drug metabolism and pharmacokinetics, it is called as ADME process, ADME. And then toxicology, safety pharmacology or toxicology. That's why it is called ADMET. ADMET screens are basically for optimizing the lead molecule which you are having, whether it is really developable or not, to find out. Those kind of test systems one need to study.
Thank you, sir.
Thank you. Yeah.
Sir, myself, Dr. K. Sivaya, sir.
Okay.
From Karaja Farms only. Sir,
Okay.
I have one small doubt regarding in autoimmune disorders, sir, especially which parameters we can evaluate, sir. So because of so many autoimmune disorders is there,
Yeah.
common parameter, which one we can measure, sir? Or particularly we have to measure psoriasis for different or rheumatoid arthritis is different?
No, no, no. See, so the thing is, in general, what happens is, what you are trying to ask is like a full-time question, which will take a
Yes,
lot
sir.
of time.
Yes, sir.
I'll try to just address it in a simple way. Most of the disease pathology mechanism at a molecular level mechanisms related to autoimmune disease are very closely linked to, you have a coin, two sides of a coin, we say, right? Heads and tails. If one side of the coin is autoimmune, the other side of the coin is cancer, oncology. Because why I am
Yes, yes.
saying this, most of the molecular mechanisms are overlapping are common. Like if your immune system is low, if somebody's immune system is low, right, what happens? He may get opportunistic diseases. Most of the times when we say immune system, it's our white blood cells, T cells, B cells, all these things, and then various mechanisms at molecular level that are involved with those mechanisms. If your immune system is down, then you end up having oncological, immune system and inflammatory. Because most of the times
Sir, inflammatory
in a cell inflammation, most of the inflammatory activities are very closely linked to your immune system. So whenever these things are on the downside, most of the times you will end up having one particular kind of cancer or other. Whereas when somebody's immune system is or people may end up having bacterial or viral infections. Like nowadays we are seeing, a lot of
It
COVID
depends,
patients.
sir. It
If
is
their immune system is not good enough, then they fall sick and they may get COVID or any other viral infection.
yes,
They
sir.
are easily prone to get infections. So one is infections, and autoimmunity is in a way if the immune system is overactive, then you will end up having arthritis, psoriasis,
Yes,
this
sir.
kind of, or ulcerative colitis or Crohn's disease, people call.
Yes, sir.
All those kind of indications will normally happen. So it's kind of a
Yes, sir. Yes, sir.
two sides of a same coin. Either you have, if it is down, people will end up having bacterial or viral infections, or even actually may get some kind of cancerous developments. And on the other side of the coin, you are dealing with the autoimmune aspect. Yeah.
Thank you, sir.
I
Thank you,
hope
sir.
it cannot be addressed so easily
Yes,
in
sir. Yes, sir. Yes, sir. It is a
1 or 2
difficult.
minutes.
It is a
We should
vast. That's why.
should
Okay,
have
sir.
taken
It's okay.
time. Yeah.
Sir.
Sir, yeah. Sir, Tippeswamy sir, you can talk now. Sir, am I audible?
Yeah, yeah.
Dr. Tippeswamy sir. He has raised his hand. Yeah, if at all any other participant want to interact with the speaker, kindly unmute your mics and
Yeah, go ahead and ask whatever.
yes, if anybody have any doubts or if anyone want to interact with the speaker, kindly unmute your mics and you can talk to him directly. Sir, Tippeswamy sir, your mic is unmute now. You can talk now directly, sir.
Ishak sir, I have a question. Sir,
Yes, please.
once again, I'm not disturbing. Sir, in other countries, foreign countries,
Yeah.
there is a industry academic interaction for drug discovery and all. There will be MOUs or some kind of understanding between industry and the academic people.
Yeah.
Why that doesn't happen in India?
Yeah, actually, so it's a very interesting question from, especially from an industry perspective. There will be some kind of things, but the only thing is, uh, especially from an Indian perspective, there are some set of what you call academic institutions which are run by government, right? CSIR laboratories, and CSC, like you have, like in Lucknow, Center for Drug Discovery, that CDRI, Research Institute, CDRI, and various you have in Hyderabad, so many ICMR institutes, in the Indian Institute of Chemical Technology and all. The problem what happens is, especially in India, I will try to address this because you asked India specific question. The thing is that the minute comes with anything comes with with government, this thing, they want, irrespective of what is their contribution, you try to approach them. The first thing, okay, whether you will give patent rights to us, how much share. A drug discovery process itself is a very lengthy, and it takes years. Yeah, and several years, a minimum 8 to 12, 8 to 10 years it will take a project from a lab scale to clinic to become a particular drug, project becoming into a drug, a reality into a drug. It takes 8 to 10 years and a lot of investments. From a, even from a international perspective, from a big pharma, anywhere between 1 billion US dollars are required. But even in India, for industry also, you need to, there are certain fixed costs, especially most of the cost of the drug discovery program happens in clinical development, where you need to pay hefty amounts to the volunteers, patients, and healthy volunteers. So most of the drug, and nowadays across different drug regulatory authorities and all, like a lot of safety, like you come regulatory authorities, whether US FDA of the our Indian regulatory are that they are not. Everything is being fixed with but international drug regulation is very strict. People don't want any kind of toxicities. First and foremost thing is the drug need to be highly safe. It should not have cardiovascular safety and other kind of regular safety. All these things are very strict. People are very strict. So without in realizing what kind of investments that will go on, all the minute Indian bureaucratic research, government research laboratories, you try to collaborate, they will try to first they'll how much whether you will give patent rights. Why they may be, but that thing, that particular aspect has changed a lot in the last 10 years. Most of the Indian drug laboratories and government institutions, where a lot of investments have been done to put up a lab scale instrumentation and all. Now they are doing everything is, it is there is no collaboration or no memorandum of understanding on the actual project. It is typically service based collaborations are there. Okay, this particular we are having something, we need to utilize this particular instrument which is available. People will do only what I am doing is nothing. I am having something, I need to test, you have facility, so we want to utilize your that kind of a service related collaborations and small time things, that those things are now slowly started happening even in India. What you are trying to say is they are these kind of things normally happen in Western countries. Those kind of things started happening, and nowadays people are having such kind of service oriented collaborations as well. But I shall tell one particular aspect. Even in US or anywhere in Europe also, uh, if normally what happens is, if let us say, I'll give you one particular example. Especially there was a few years back, there was a particular research work that has been carried out at Harvard Medical School. So it is like related to diabetes related, metabolic disorders related. One particular mechanism which came in news at that point of time. I don't want to for highlight exactly tell this thing, but a particular thing has been said to be very prominent, which is going to be change, especially from an endocrinology perspective, a new drug target, new mechanism which has been found out. So what they did is GSK, they bought that entire intellectual property and technology related to that. They just went and bought that everything in for few hundred millions. They bought it, and then that there is no longer role. They just took over the rights of that particular program from Harvard Medical School, and they ended up, unfortunately, that whole program ended up becoming a damn squib. What otherwise people thought an excellent mechanism or something, highly innovative thing, it turned out to be మనం అంటాం కదా తుస్సు అన్నట్టుగా డ్యామ్, it turned out to be a damn squib program. And unfortunately GSK spent some 500 millions they gave to, but most of the times what happens is that most of these deals, they happen if everything goes well, you will, the total deal value is 500 million, but initially they don't pay that much. They will give you, okay, we are taking your novel idea and intellectual property. Initially we are paying $30 million or you $50 million, something like that. If this program successfully becomes successful and it gives us a drug candidate, then we will pay you total over a period of time. Like this is called as one-time payment is one thing. As the program is progressing, they will be doing additional payments. Such kind of collaborations people will have. It's called out licensing fees. These are typically from a business perspective, some of the terminologies people use.
Yes, thank you.
So for the benefit of students, I would like to highlight one thing to all the faculty at your college and also to the management. It's one kind of medical intervention is very much required. So for that kind of an extraordinary situation, drug repurposing has become very interesting, quick solution thing. But that is not the mainstay approach. Most of the times, like otherwise, there are so many diseases, so many pathological diseases, and so much of thing, and regular drug discovery, it will people will as long as people are there, diseases are there, and some kind of intervention or thing need to be discovered, and these things will keep on happening. Okay.
Yes, sir. Thank you. If any speakers, if anyone of the audience, if you want to interact with the speaker, kindly unmute your mics and you can interact with the speaker directly. No more questions from the audience, sir. Thank you so much for your valuable time, sir. Thank you so much for this wonderful session, information session.
Yeah, thank you very much. I hope I tried my level best to give you all guys at academic level, try to give as detailed methodologies normally people in industry adapt, try to uh, importance, all those things. I try to highlight those things. I hope you people appreciate and understand. I hope it will be useful for all students, especially, because based on certain information, they will be able to better, especially for bachelor's level students, what kind of specialization they should be taking at a master's level and all. I hope it will be useful in some.
Yes, sir. Definitely, sir. Definitely, sir. Thank you so much.
Not you tried, Dr. Srinivas. You really did a good job and really is very useful information. So not level best is 100%. You clarified our students, our participants. Thank you, Dr. Sinu.
Okay, thank you very much.
Thank you, sir. With the permission of the principal and organizing secretaries, we will like to felicitate Dr. Srinivasan sir virtually. Srinivasan, kindly accept our virtual felicitation.
Okay.
Thank you so much, sir. Thank you so much for your valuable presentation once again. I hope and we will see few more collaborations with you in future. Thank you, sir.
Yeah, thank you very much. I was like a bit, worried as we started around 2:30. At that time, it was there is a very bright sunlight, and in the last 30 minutes outside in Mumbai, situation suddenly changed. It is raining very heavily. I just thought in between power goes off or something, some problem. But thankfully, everything is fine.
Yes, sir.
Thing went well.
Thank you, sir. I request Dr. Shivaya to kindly propose a formal vote of thanks.
Thank you, Isaac sir. It is an honor to have been to offer a vote of thanks on the occasion of International Webinar on Pharmacological Screening Methods today. I am very heartily gratitude to speaker Dr. Srinivas Gullapalli Garu for accepting and sharing of on preclinical drug discovery research perspective. Sir, well concluded, sir. Thank you. I am thankful to our chairman, Dr. Santiramadu Garu, Director Ashok Kumar Garu for giving opportunity. I am very much thankful to our beloved principal, Dr. C. Madhusudan Sethi, for continuous support and advices which have greatly helped towards the success of this webinar. And my thanks goes to Dr. M. Srinivasul Garu for obtaining good speakers and initiation of this webinar. And I am very thank you to our the program coordinator, the Ravikumar Garu. And I am very thank you to Dr. Ishak sir for hosting website and making the flyer. I am sincerely HOD of the and faculty of Santiram College of Pharmacy for providing all possible logistics towards organizing this event. Ultimately, the students make the bond all companionship. I'm very, very thankful to all our students for to attending this program. Very, very thank you, sir.
Thank you, Dr. Shivaya. So that's all for today's session. Thank you so much for being with us today. A small note that tomorrow, again, we are going to meet once again, even though it is a Sunday. By 10:30 a.m., we are going to have a wonderful session with Dr. Yashoda Krishna sir from Kenya. And afternoon by tomorrow by 2:30, we will be having a session by Dr. Tippeswamy sir, who is joining us from Saudi Arabia. So this is for information. Thank you so much. Tomorrow we will meet once again. Thank you, one and all.
Okay, thank you very much for all
Thank you,
you
sir.
guys from Santiramadu College. And once again, I thank all faculty and my beloved friend Madhusudan Chetty, Dr. Chetty, and also to the management of the college for providing this opportunity to interact with all you guys. And good luck for other programs as well.
Thank you, sir. Thank you, sir. Thank you, guys. Signing it off. Thank you.